Okay, so let's talk about, hi everybody, here, I'll do my best. All right, let's talk about first the central dogma, right? So I spent some time talking about the central dogma of DNA. And what does that really mean? What's the central dogma mean? Huh? The principle? Where's my drawing? Oh, it's over here. So the central dogma is really talking about the flow of genetic information inside of our cells. We haven't broken down into cells yet, but we're talking about DNA. So the flow of genetic information, right? So we start off with DNA. What do we go to? RNA and proteins. But then what's after proteins? Traits. That's annoying. We'll have to close that. So DNA to RNA to protein. So we have to take DNA and turn it into RNA language. And what is that process called? We are transcribing. Does that make sense? So we have transcription. So now what do we do with the RNA language? The RNA language then needs to be translated into proteins. So guys, we've talked about protein structure at great length. And I remember introducing the primary structure of proteins and saying to you guys, it's determined by DNA. So that's where we are right now. And these proteins are what determine whatever traits that it's coding for, okay? Like your genetic traits that you think about, your hair color, your eye color, all of those things. But really everything in your body is run by proteins. So this is really important. So before I start like drawing my picture and showing you how all of this goes, I want to just talk about the differences between DNA and RNA. Who can tell me?
It's single strand RNA and DNA is double helix.
So we have a single strand. Yes. DNA is double helix, double strand. What else?
RNA has a
We have a uracil. What do we have here? So RNA, there is no T, right? And then I heard somebody say there's ribose and deoxyribose, okay? So these are important things we need to consider when we're talking about transcription and translation. All right, so what I've done here, I've got all my colors out. I started drawing, so I drew our nucleus. What's inside of the nucleus? DNA. What process are we going to be talking about here? Right, so we have transcription. And I know when I give you these PowerPoints and I can't really articulate as well as I want to and show you how these things work, that it can feel really overwhelming. And my goal is to make this as simple for you as possible. So you can notice that I have 5' to 3' and a 3' to 5'. Which one is our template strand? Yeah, this is our template strand. How is mRNA made? What direction? 5 to 3. It needs to be made 5' to 3'. Okay, so that directionality, again, is important. So you can see that I kind of started showing that this is unwinding. I haven't spent a lot of time. That's not true. What is this thing right here? The Tata box. What is that? Promoter, right? So we know, our enzymes know where to attach because what ends up happening is we have a bunch of different proteins that attach here and tell the RNA polymerase II, hey, start here, okay? So our Tata box, why do we think TA is the one, a bunch of TATAs? It is. It's not the start codon. It only has 2 hydrogen bonds that need to be broken as opposed to the 3, so it's a little bit easier. All right, so we have our Tata box, and you can see I made a code here. What is the name of this enzyme? Yeah, RNA. Do you guys remember what's the name of the one that, the polymerase that works in DNA replication? What's that name? DNA polymerase. Well, yeah, no, that's DNA polymerase, but we're making RNA, so it's RNA polymerase, right? All right, so this is polymerase II. So we see that our polymerase is working in this direction. All right, let me get my green that I'm using for my mRNA. So my little green thing is the mRNA being made, and let's all do transcription together. What letter goes next to this C?
G.
G. What do we have here?
G.
No T's.
No, U.
U. What's here?
U. G.
G.
C. A. C. U.
Okay, you guys just did transcription.
C.
That's transcription. See how easy that is? Okay. So transcription is occurring, and I want to make some major points that are very important. The thing that gets made here is called the pre-mRNA. It needs to be modified, okay? So who can remember the things that I said happen before the mRNA leaves the nucleus? What's one really important one? Splicing of introns. So do you guys remember what introns are? What are they? Non-coding DNA. We have a lot of non-coding DNA. Now, there's a lot of theories out there. They used to call it junk DNA, and scientists are very leery on calling it junk DNA anymore. There is a better understanding, or there isn't a huge understanding, but there is more of a hypothesis that this is just DNA that hasn't been turned on yet, right? We have coding sections in our DNA that we don't know what it's there for, and we don't know why it's there. There are some implications that it's viral DNA, because if you know how viruses work, viruses inject their DNA and kind of incorporate into ours. So like over time, it could be some excess viral DNA, but we have non-coding DNA. So non-coding is introns, coding is what we call the exons. So I'm just going to write that here so you guys know. That's our coding regions. But we also call that what? Yeah, a gene, okay? A gene codes for a protein, okay? So DNA codes for a protein. These genes code for a protein. We have different genes that code for different things. All right, so we splice out the introns. So our mRNA gets shorter. Let me just draw a little shorter mRNA here because we're going to splice out the garbage. We're just going to, whatever. Let's see, what's the word am I trying? I'm just going to put a little code here. Okay. What else do I need to add on? This is our 5' side. 3' side. What else do we need to add on? There's 2 more things that get done to this RNA before it's ready to leave. Yep. We have a 5' cap. 5' cap. What is that? What does that do? Yeah, protects it from enzymes. And then what's the last thing that we do?
The 3' tail.
The poly-A tail. Now, it's actually really cool that this happens, okay? As soon as this mRNA leaves the comfort, the safety of the nucleus.
Is it always the same amount of A's?
No, I just random poly-A. I just put a bunch on there. It depends. It can be different sizes depending on the size of the gene. Because these guys serve as protection. As soon as this mRNA comes out, there's going to be lysosomes, there's going to be enzymes, there's going to be all sorts of things that attack it. This 5' cap protects it from enzymes, and what's going to happen is just enzymes are going to come in and just start cutting those off. And what happens is the enzymes cut off the coding region. Yeah, the DNA doesn't work. So the longer the poly-A tail, the longer the code, because it takes longer to translate it. Does that make sense? All right, so let me draw my little mRNA out here. Actually, I'm going to do this to make a point. All right, so where's my guy going? Yeah, let's make my ribosome pink. Okay. I like to color code everything. So the ribosome we learned has how many parts to it?
2 parts.
Yeah, we have a large subunit and a small subunit. And then I have to draw some other things because I really like to draw. What am I drawing? Yeah, so we have another type of RNA called tRNA. And what do these tRNAs carry? They carry amino acids. So Is this starting to make sense? Like, these guys carry, I'm going to represent them by different colors. I'm going to put AA right here. We'll do a pink one because that's pretty. And we'll make it a star shape just because whatever. All right. So we have our tRNAs. And what do the tRNAs have here? What are these things called? Yes. Why do they have anticodons?
So that they can stay.
They can attach to the mRNA, right? They can attach to the mRNA so they can help put the proteins together. All right? So I've drawn these random tRNAs. I'm missing some spots for my ribosome. So the ribosome has 3 spots. E, P, and A. Do we remember this part? Okay. So the ribosome is going to come around and attach to the mRNA. Okay? And it's got 3 spots. This is the exit spot. This is when our RNAs leave empty. Our tRNAs are going to leave. Here's the spot. Oh, let's keep the colors the same. Sorry, guys. What's going to happen in this spot? What happens right here? What are we building here? This is where the proteins get made. Okay? So some of you guys may be like, Norige, you're crazy. This is too much. It's not. Trust me. You guys are going to draw this, and you're going to love it, and it's going to be fun. And we're going to have a pizza party when you all get it. All right. We're not going to have a pizza party. I can't afford that much pizza. All right. So, and then we have another tRNA landing here. So, this is our landing site. Right? G, G, C. And then this one's gone already. This is our landing site. Okay? Then what happens is the ribosome moves. It goes here. The proteins get put together. Dehydration synthesis is occurring here to make these peptide bonds, right? So we have this growing chain of amino acids, which is the primary structure of proteins, and then it leaves and goes around, and it's going to go find another amino acid because the amino acids are all hanging out, and they're going to just keep working in this way. That occurring right here is translation. So I took some time in lecture to show you guys how to use universal genetic code. I'm not going to spend a significant amount of time on it today in class. I do, however, have an activity to give you practice on how to do it. It's not a grade, but if you want it, make sure you grab it before you leave. Go over it. I'll go over it at the beginning of next class. So, am I missing anything here? Are there questions here? We have our DNA, our promoter region, RNA polymerase II, mRNA being made, splice the introns out, put a 5' cap, poly A tail, goes out to the ribosome. tRNAs bring these amino acids to make a protein. How do we know when to stop, by the way? What do we have? How does this know when to stop?
It's going to be a release.
Yeah, if you guys take a look at your universal genetic code, if you take a look at UGA, what's it say next to it?
Stop.
Yes, that mRNA code UGA and several others tell the amino acids or the ribosome this is we're going to stop. The ribosome detaches, protein goes where it's supposed to go, and then we're done. Okay? This is a lot. So, because it's a lot, I'm going to stop this here.